with a dihybrid cross. All right, as soon as I launch the YouTube page, I'll share where you should be and what we're going to do today. Okay.
The things you're into are always changing, but what you pay for internet shouldn't. Switch to Verizon Home.
Okay, that just, because it was YouTube, it just launched itself. Okay. All right, so trying to get the GoGuardian up. So many things, so many things. All right, that's done. Try to do all these things so that it's not popping up while I'm sharing. Okay. All right, so in Canvas, this is where we should be. And I want to point out, we're going to be looking at the different types of inheritance. Today, in particular, we're going to look at sex-linked traits, and then we'll look at polygenic inheritance. But we're going to look at all of these different, there's all these different types of characteristics. So I think what I'm going to do is show you the video first and then give you time to finish the first worksheet and then come back and read this page on your own. So we're going to look at, so let's look at this first column first. We're going to look at sex-linked traits and how they're located. Okay, so if you look up here, this is the information we're going to look at in the video today. The fact that you have the mapping of we have 23 chromosomes, but only one set of them is set out to determine whether you're male or female. But on those chromosomes, there are certain characteristics. Yes, that's the genetics module. It is definitely the genetics module. Types of inheritance, genetics module. It's published. You look at it as a student. I published the whole unit. All right, so we're looking at this today. And like I said, we're going to come back and look at all of these, but today we're just going to focus on the sex-linked traits, and keeping in mind that each person has 2 chromosomes that kind of split during meiosis. So one goes in one cell and the other goes in the other. And in the case of sex cells or gametes, you're either giving up, and if you're a female, you only have you have two X chromosomes to give. So each cell is going to have to get an X chromosome. But if you're male, you can give an X or a Y chromosome. So I'm going to go ahead and show this video, and I'm going to pause it. So be prepared to write something down, because I'm going to pause it when it gets to the part where she has worked out the Punnett square for this, because this is what you're going to have to do. We are going to have a test. I'm not going to see y'all until Tuesday because of the holiday weekend. So they shifted the schedule so that there's no Wednesday next week. So I will see you on Tuesday, and we're going to have a little quiz. And quizzes in this case are a paragraph that gives you a description, and you have to draw the Punnett square and answer the questions that they present. And it's usually, what is the genotype? What is the phenotype? What are the results? All right, so we're going to watch this video. And again, have a paper and pencil out, because I am going to pause it at a certain point for you to write down what they have on the screen.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cells that help stop us from bleeding. They help our blood to clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets, and therefore the ability of blood to clot. With hemophilia, even a small cut could be dangerous because the bleeding can be continuous. There are now many good treatments for the symptoms of hemophilia that have greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it is different from general Mendelian genetic problems. With sex-linked traits, we still use the terms dominant and recessive for alleles, except this time, those alleles are on sex chromosomes. This is the case with sex-linked traits. What is a sex chromosome? Well, first take a look at this karyotype, which shows a human's chromosomes. This karyotype has 40...
All right, we're going to be using the term karyotype later on. We haven't come across it yet, but it is on your vocabulary list. Karyotype is just a picture of all the chromosomes where they lay out all of your chromosomes like a map, and they number them. So in case of humans, there are 23 pairs, but the only pair that they're going to look at are the ones that are boxed down here. The 23rd pair is called the X and the Y chromosome. So karyotype is a word that we're going to have to know, but not right now.
46 chromosomes arranged here in 23 pairs. 46 is the general number of chromosomes in humans, but as we mentioned in our karyotype video, individuals can have more or fewer than 46. Chromosomes are made up of DNA and protein. They contain your genes. 23 of this person's chromosomes came from an egg cell, and 23 of this person's chromosomes came from a sperm cell. The last 2 of the 46 chromosomes in this karyotype are called sex chromosomes. The sex chromosomes are commonly called X or Y chromosomes, but X and Y have nothing to do with the shape of the chromosome. The reason for the X and Y names is really interesting. Check out our further reading to learn more. All humans have at least one X chromosome. In most cases, females have a combination of XX and males have a combination of XY, but individuals can have additional or fewer sex chromosomes. We're focusing on sex chromosomes in this video because we're going to show how to do Punnett squares with traits on these chromosomes. In the case of Punnett squares with sex-linked traits, the traits being tracked tend to be on the X chromosome and not the Y chromosome. The X chromosome is much larger than the Y chromosome and contains more genes than the Y chromosome, so technically this video will be about X-linked traits, but we do want you to know there are Y-linked traits too. Referring back to the beginning, when we mentioned the disorder hemophilia, hemophilia is a sex-linked recessive disorder carried on the X chromosome. So focusing on that, let's learn how to solve a Punnett square that involves this trait. We will use the capital letter H to represent an allele for not having hemophilia, and a lowercase letter h to represent an allele for having hemophilia. Why? We're going to do that because we mentioned hemophilia is a recessive sex-linked disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the X chromosome as a superscript, like an exponent. Let me explain what I mean by that. Let's consider a female individual shown with XX here. If the genotype is X big H, or X big H, X little h, this female will not have hemophilia. Why? Because as long as there is at least one dominant allele, that dominating allele will be what shows in this trait. So, no hemophilia. Hemophilia is a sex-linked recessive disorder. However, the X big H, X little h genotype would be a carrier, meaning while the person wouldn't have hemophilia, they would be carrying the recessive allele. The only way for this particular female to have hemophilia would be the genotype X little h, because only when there is no dominant present will that recessive show up, at least in this type of trait. Let's consider a male individual, shown with XY here. X big HY would be a genotype of a male without hemophilia. Notice how I didn't put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If this male had the genotype X little hy, then this individual would have hemophilia. Since there aren't two X chromosomes for this person, either this person has hemophilia or doesn't. Let's try a Punnett square problem with this trait. Let's say these two people here both do not have hemophilia, although the female is a carrier of hemophilia. If they together have a biological child, what is the percent chance of the child having hemophilia? Also give genotype and phenotype ratios. Step 1. Always determine the genotypes of the parents first. The female must be X big H, X little h. Why? The Punnett square problem says the individual doesn't have hemophilia, but is a carrier. That means this heterozygous genotype. It says the male does not have hemophilia, and so the genotype must be X big HY. If it was lowercase h, this male would have hemophilia. Step 2, place one parent's genotype on the top, outside the square like this. Place the other parent's genotype on the left, outside of the square, like this. Step 3, fill in the square. For formatting...
All right, I'm going to pause this and go back so you can write the question. And then I'm going to come back and let you just write down the results. So, oops. Okay, right there. All right, write this question down. What would be this percent chance of a biological child that has hemophilia if the male does not have hemophilia, but the mother is a carrier? Okay, right? That's the question. That's the situation that you have. What would be the percent about that a percent chance? And remember, all of this is possibilities. When we do a Punnett square, it's about probabilities, or how much out of 100%, what percentage of the children could possibly have that trait? Okay, because offspring are random when they're actually born, not on paper, but in actuality. So you don't know which egg and sperm are going to create which offspring. So when we look at a Punnett square, it's about possibilities. So what is the percent possibility of a biological child having hemophilia if, and then you have to write down, the father does not have hemophilia and the mother is a carrier. She does not have hemophilia, but is a carrier. All right, so that's the problem. And then I'll skip back to where she's setting up the Punnett square. Again, this is the problem. And I'm asking you to write this down as an example for you, because and going forward, this is how the problems will be set up. It'll be a paragraph that explains a situation, what the characteristics are of the two parents, and then you will take the information from the paragraph and create a Punnett square and create a result table. Okay, so that's where she was. All right, so and I'll pause it again when
Step
she's
3,
finished.
fill in the square. For formatting purposes, place X chromosomes before Y. You also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. And the phenotype ratio, remember that these are the traits, can be written out like this. There's a 75% chance that a child will be born without hemophilia.
Okay. So copy the Punnett square. Notice they put the mother across the top so that you have your X and X. And because, like she said, she does not have hemophilia, that capital H represents no allele for hemophilia, but the lowercase h represents that they have an allele for hemophilia. But it is recessive. So that means that it can be masked. So she does not have hemophilia, but she's a carrier. Dad, on the other hand, has a normal allele on his X chromosome. Letting someone in. And there is no allele for this trait on the Y chromosome. So the only thing that can be expressed is the normal gene for hemophilia. And again, this has to do with your ability to clot, with your blood to clot. So your body tells you this is what you need to be able to do. It creates a protein that clots the blood when you breathe. So for those of you who don't know, that hemophilia is what they call the bleeding disease. You do not have control because you're not going to clot. You will bleed continuously and you could bleed to death. Now, these days, they have drugs that they can administer quickly. As they said in the past, people just bled to death, or they tried not to have offspring that are going to have that trait. All right. Now, the question again is going to ask you to read the boxes. You have to be able to read what's all the letters that you put in the box. So genotype is what genes are in the boxes, and you have to have 2 genes in every box. And so when we look at this one, we have one box that has the 2 capital H's on the X chromosome. We have one box. Actually, we have 2 boxes. We have actually one box that has a capital H and a lowercase h. So we have 2 females, and both females, because we have X and X, we have 2 females, and they're going to be normal for hemophilia. On the other hand, we have an X and a Y with a capital H, and that's a male, and he's going to be normal. And then we have an X and a Y with a lowercase h, and because there's nothing to hide that lowercase h, this child is going to have hemophilia. All right, so phenotype, 3 of them will have hemophilia, will have no hemophilia, will be normal, and 1 will have hemophilia. And then the last thing you can do is represent it as a percentage, because that's what the question asked you. What is the percentage? And it will be 75% chance that the child will be born with hemophilia, without hemophilia, sorry, and 25% chance that they will. Okay, so you have to do all of these steps and then lastly answer the question. The question was, what was the percentage? So tell the genotype, tell the phenotype, and then give the percentage. All right, so hopefully you copied all of that down. Well, I'll give another minute in case you were just listening while I was talking. And then I'll start the video back. And I gave you this because this is an example of how you are to answer the question.
And a 25% chance that a child would be born with hemophilia. Remember, like all Punnett squares, these are representing probabilities. So because it's a probability and not necessarily the exact outcome, they could, for example, have quadruplets that all do not have hemophilia, or they could have quadruplets that all have hemophilia. The probability of the latter is less likely, but it's still possible. 5 things to keep in mind when you are working on these kind of Punnett squares. Number 1, you do not want to just assume a trait is a sex-linked trait, because many traits are not sex-linked. More traits are found on the autosomes, actually. Autosomes are all the chromosomes that are not sex chromosomes. When you first started practicing with Punnett squares, it's likely those were problems that were not sex-linked, and thus the sex chromosomes of the parents would be irrelevant on the square. So never assume. The problem should indicate it's a sex-linked trait in some way. Number 2, did you notice how the child in our example that would have hemophilia was XY? Sex-linked recessive traits are more common in XY genotypes compared to XX because XY has only one X chromosome. For example, some forms of color blindness are sex-linked recessive. It is more common in males. When arranging a pedigree that is tracking a sex-linked recessive trait, it can be common to see a lot of shaded squares in the pedigree, representing males that have the sex-linked recessive trait. You can learn more about pedigrees in our pedigree video. Number 3, you may be wondering, our example was a sex-linked recessive trait. But can there be a sex-linked dominant trait? Yes, a sex-linked dominant trait means it would only take one dominant allele for the individual to have the trait. Let's use a letter D to illustrate a hypothetical sex-linked dominant trait. If it was a sex-linked dominant trait, these 2 female genotypes would have the trait, because again, it only takes one capital letter, a dominant allele, to have this dominant trait. This female would not have the sex-linked dominant trait. This male would have the dominant sex-linked trait, and this male would not. Number 4, because we talked about the inheritance of the disorder hemophilia, and we also mentioned color blindness, we do want to make sure you don't leave with a misconception. Not all disorders that have a genetic component follow a one-gene kind of trait. In fact, many don't. An example? We both talk about how we developed preeclampsia, a disorder that occurs during pregnancy or the postpartum period that can be life-threatening to those that develop it. While current research is studying genes that could play a role in developing preeclampsia, like genes that impact the placenta or genes that involve the vascular endothelium, the cause of preeclampsia is overall still not well understood. Many disorders, though, that have a genetic component can have multiple genes interacting together and wouldn't be something that you could place in a general Punnett square. And these disorders can have external factors as well that are separate from genetics. Number 5. Our example was for humans. Humans are animals, but many animals do not have X and Y sex chromosomes. For example, birds, where it's Z and W. Or some animals might have X and Y sex chromosomes, but maybe they typically have 10 of them, like the platypus. More in our description. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. All right. So coming back to Canvas, the other one that they mentioned was polygenic. So sometimes you have more than one gene to represent a particular trait, such as eye color. So we're going to look at that one next time. So like I promised, I'm going to give you some time to get that other worksheet submitted. If you finish working that worksheet, come back to this page and go through all of these, because this is what we're going to do next. We're going to look at complete dominance. We're going to look at incomplete dominance. We're going to look at co-dominant, which means that instead of getting red or white, we get pink. We just looked at sex linked, and then we're going to look at lethal alleles. So make sure you come back and look at these and just go through this page for a practice. All right, so get that worksheet done. Y'all are taking way too long with that. And let's see make sure that you, all right, so I'm talking about this one. I think there's only one or two of you that have not done the other. All right. So this one needs to be in, and then take your time and read that page, because there will be a worksheet. So again, it's going to get more complicated. This one is what we called a dihybrid cross. But when we look at polygenics, then you're going to have multiple alleles to look at in a box. So this box has 16, the box doubles when we look at, say, 3 alleles together. All right, so when we do look at skin type and eye color, that's what's going to happen. So make sure you get this done. If you have a question and you don't understand this, please say so, and I will try to re-explain it to you. All right. Like I said, I only had one person who had turned in this worksheet, so everybody else owes me this worksheet. We're trying not to get behind. It's too early in the semester to start falling behind. And like I said, I'm going to start leaving the late designation on the assignments if they're late. This one is bordering on late. After today, it's late. Yeah, because it was, okay, it was due Friday. So after today, it's late. We don't want to start having assignments start to pile up on us. Excuse me. Again, if you're finished the worksheet, get that turned in, go to the Canvas page and read on types of inheritance. Yes. Well, on this worksheet, it depends on how badly you messed up. Usually, unless you totally messed it up, I usually just take the 1 or 2 points off for whichever one you got wrong. But um, where's my worksheet? They all of the at the bottom, they have to add up to always remember there's 2 genes in every box, and then you're reading what that box means as far as the step 2 is concerned. The third part, all of the numbers should add up to 16 because there are 16 boxes. So you'll know before you turn it in whether you've messed it up or not. And you're just reading, you're looking at each box. And what I typically do when I do have boxes like that is I cross out the one that's already been used. So there should not be a reason. There's a way to double check yourself. There should not be a reason to mess up so badly that you have to redo it again. You might have one mistake, but not totally mess it up. And that's the neat part about this is, and actually let me show you the Canva page again that I had. Okay, this is the same thing as the box that you're working on. Okay, and all they're asking, and it may look better this way because each box is colored, yours is not. Um, individually colored like that, but it's the same thing. Okay, we're looking at two different traits, yellow and green, wrinkled and round. But when you have a dihybrid, you're splitting them out. So we're at the top, we had the Ys together and the Rs together. During mitosis, they're going to separate. So one chromosome will have its Y and its R, and the other chromosome will have a Y and an R, and they're going to different boxes or different cells. And then just take your time and count, because that's all you're doing with that bottom one. You're looking, how many times are you going to see capital Y, capital Y, capital R, capital R? So our probability changes from when we're just looking at two traits. Now we're looking at, when we're looking at one trait, the monohybrid, and then we start looking at two. And it's going to change again when we start looking at three. So again, all you're doing on this page is saying one. You can say 1 out of 16, 2 out of 16, 3, but all the numbers should add up to 16. If it doesn't add up to 16, then you missed the box. And you can't miss a box because the exact outcomes are already listed for you. These are all the possible outcomes that you can get with this cross. All you're doing is counting them. Excuse me a minute, I'll be right back. Yes, I can. Okay, give me a minute. They are correct. So Kendall, yes, you're good. All right, Michaela, do you know what we're supposed to be doing?
I'm working on the worksheet.
Okay. And I say that because when I'm looking at Go Guardian, it doesn't have you engaged in anything. So just checking. Just checking. Jacoby, you too. Hopefully you are working on yours. Thank you. All right, so again, hopefully by the end of this lesson, everybody needs to have this worksheet uploaded. It's not that long a worksheet. It should be uploaded. And I want to be able to, so in the next 10 minutes, I'm going to explain the polygenic one. Uh, Okay, I want to take a minute to show you the worksheet that went with today's lesson. So you might have to refresh, but it's going to be on your homepage on To Do. It's not a module. And it'll be the Sex Links Genetics Calico Cat. All right, and when you click on it, it should look like this. So there's only 4 questions, but with the first question, they give you, again, the information of what you need to draw who is dominant, who is co-dominant. Okay, code color for the cat is sex-linked, but it's also co-dominant. So again, you'll need to go read that section on Canvas as to what we mean by co-dominant. Now you see this cat, it's got multiple colors. It's not black, it's not white, it's not brown, it's got multiple colors on it. So that's a calico cat. So you are going to have to place the letters in the box according to what they tell you. So this first one, so these are all the possibilities up here at the top for the male, for the female, and for the male. All right, then it asks you, a black male is crossed with an orange female. So you look up here at the top, it tells you that for black, it has to be X with a capital B and a Y. So that's going to be what they wrote at the top. On this one, they placed the male across the top. Then it tells you that it's crossed with an orange female. So if you come back up here to the key, it tells you that an orange female is X with a capital R and X with a capital R. So that's what you're going to put on the side. So they've already done that part for you. So that's the female. So that's where they got those letters from. All you have to do is complete the cross, put it in the box, and then answer the question. How many kittens are orange males? How many are calico females? And note those are your choices. Okay. After that, you have to draw the box. So you have to, for 3, 2, 3, and 4, you will have to draw the Punnett square based on the information that they give you in the question. And then do the same thing. Come up with an answer that show the phenotypes and draw the Punnett square and list the phenotypes and proportions of each. So all you're doing again is, once you complete the box, tell us what the box is saying. Okay. Question number 3, the same thing. All of them say draw the Punnett square based on the information that they give you, and then draw a Punnett square and list the phenotype and proportions. Okay. All right, so that went with today's video. So like I said, y'all are starting to fall behind because you're taking all day to do one worksheet. But I put this up because I'm trying to keep you in pace with the other class. Okay, so it's there. If you finish with the other one, please start on this one. This one's not due till Tuesday. And we'll, again, you should go back and look at what we mean by codominant. So you were supposed to look at the module. And so there's a lot of things you got to do. Look at the module, and we'll talk about, on Tuesday, we'll talk more about codominance. And where am I? Types of inheritance, that's where we are. Types of inheritance. I can get it to come up. Okay, so so that worksheet deals with not only sex linked, but codominance. So please read up on that. don't think I did attendance. All right, give me 1 second to look at that. Okay. Your boxes are drawn correctly, but in the case of the first two, you're going to have some zeros. So look at it again. You shouldn't have yeah, yeah, look at your results again on those first two. And hold on. No, they're not. Okay, this first box, you have a, it shouldn't be two lowercase h's. It should be a capital H and a lowercase h in that bottom box. Because you have a capital H on the left and you have a small h on the top. So that box on the bottom should also have, yeah, fix both your bottom boxes. There you go. And then the next one. Yeah, that's right. All right, the next A's. Okay, your bottom left box, because you should be crossing a small a and a small a. So that's what should be in that box. The other ones are correct. Yeah. All right, so now read your results. That one is fine. Read your results now and it'll be correct. Wait a sec. Okay, wait a second. You got, okay, your second box with the A's, your top box is not correct. It should be a capital A and a small a. Yeah. Okay, now read the boxes. All right. So yeah, it is very important that you all do the cross properly, because if you don't do the cross properly, you could read it right, but it'd be wrong. Okay, so do a double check. Take your fingers and drag the letters down with your fingers so that you know that you're putting the right letters in the box. And then once you put the right letters in the box, then you're going to have, then you definitely will have the correct results. Uh-huh. Yeah. Okay, you got it. Okay. All right, you're good. All right, so again, once you have submitted this worksheet, look at the one on the Calico Cat. And again, Calico Cat, it's not showing one color or the other color. It's showing all the colors because they are all dominant. So they have co-dominance, means that they are all expressed. All right, just a heads up, we got about 4 minutes left to class, so please make sure that you submit this today.